By now you have gathered enough knowledge about the genes that are present inside an individual. Now, the genetic composition of an organism is decided by the DNA that is present inside it, or in other words, I can put it that the base pairs which are present inside the DNA or the base pair arrangement of an individual decides its genetic information. So, the DNA is basically carrying the genetic information, and you must have observed that every individual is having a different genetic constitution, it varies. So, the DNA is also going to be different. So, this in a way had a very serious implication all over around us, and not just serious, this is fascinating, more of it is fascinating, and that led to the quest of finding out what human genetic makeup would be like. Okay. Now, once the quest started, the scientists who were studying about the genetic makeup of individuals, they started looking into the human genome. Now, what does genome mean? I don't recall whether we have done this term earlier. If we haven't, I am going to repeat it, that genome refers to haploid DNA content of an organism. That is its genome. Haploid DNA content. It could be a haploid organism, it could be a diploid organism, it could be a triploid organism, but the genome, wherever it is referred, it will refer to haploid DNA content of the organism. Now, you know that humans have how many base pairs? They have 6.6 into 10 power 9, that is 6.6 billion base pairs in their entire chromosomal composition. If the DNA is to be spread out and counted, how many base pairs are there? It is 6.6 into 10 power 9 base pairs are present. And as we are talking that we have to see the genome, so we will seek 3.3 into 10 power 9 base pairs. Now, the quest that had started way back in 1990s took almost 13 years to complete the entire coding, or you see the entire getting to a solution what these base pairs are. Okay, that is what Human Genome Project is. It is a mammoth project. Many countries have contributed into it. Billions of dollars have been invested in this project. In 2003 it got completed. In 1990s it began, and the quest was for finding out the 3.3 billion base pairs which are present in the human genetic composition. That means in individual cell there are as many as these many base pairs. The haploid content of the DNA was to be identified, then coded, and then presented, and then the information had to be stored as well. That part was covered in the Human Genome Project. Now, you get an idea what Human Genome Project is. The mammoth project which aimed at finding out these many base pairs using various methodologies. We talk about methodologies, but before that we have to talk about the goals of HGP. It is abbreviated as HGP, that is Human Genome Project. The developed world countries like Japan, Germany, US, UK, they contributed into this, and with their manpower, which was intelligent manpower, and apart from that they contributed with their economic strength as well in terms of money. So, they invested both manpower and money in this Human Genome Project with the goals which were, first of all, they had to identify as many as 20,000 to 25,000 approximately. There are these many genes in human genome. So, this approximate value of the genes had to be identified. All genes which are present in the genome which number to this much value, they had to be identified in this Human Genome Project. Second aim was to find out the 3.3 billion base pairs, what they are. They were to be coded. Okay, they were to be found out that how many base pairs are there and what kind of base pairs are there. Then third aim was to store this information. storing the information that was collected out of this in various databases, which coined the field in biology known as bioinformatics. Now you see, this itself is a huge number, 3.3 billion, almost half the population of human population of the world is this much. So you can imagine finding out the coding sequences for these many base pairs and then storing it. It required a lot of databases, many these IT field people had to be involved using their knowledge about supercomputers. So it gave a new field that is known as bioinformatics because it aimed at storing the information. Apart from that, there was a goal that transfer of information to the related industries, other industries was also one of the goals, okay. Industries and fields, transfer of info, that was one of the goals. And lastly, there was addressing ELSI. What is ELSI? ELSI is an abbreviation for ethical, legal and social issues. Now this project involves coding something which is related to human beings. It has many implications and many uses afterwards, as we had talked that the fourth goal was transfer of information, whatever is gathered, to other industries whichever required it. So there was an ethical issue. Ethics came into scenario, there were legal issues, those were to be addressed by this Human Genome Project. There were social issues, all these issues they were to be addressed well in accord with whatever they asked for, so that this project is in totality a success. So these are the goals of the Human Genome Project, what they are seeking, what they are looking for, why they started looking for it and what they have to address when they are doing this project. This is the basic introduction that you need to know about Human Genome Project. So we are done with the introduction. Next lesson is about the various methodologies which were involved in coding out that sequence and finding out what all sequences are there, which is in the next